Record.
Okay, very sorry for all the delays. It's as a result of the issues, concerning network and all kinds of things. Anyway, we're going to look at this particular topic today. Yesterday, we were able to successfully do our CA test, and I told you that what we have done until last week was actually supposed to cover the first part of the two courses. Now, what we are doing, we are entering the second part, which is supposed to be mainly the second course, which is molecular genetics and biotechnology. So, in today's class, we are going to be looking at molecular genetic tools. What we did previously is to look at basic genetics and then look at an introduction to molecular genetics, and then we took time to look at genome maintenance, which is the process of DNA replication. Then we look at genome expression, where we look at DNA transcription, we look at RNA translation, or what we simply describe as the process of protein synthesis. And we concluded by looking at the genetic code and the processes of mutations, and we saw the different types of mutation. We saw the characteristics of the genetic code and some of the very specific issues surrounding the genetic code, where we say that the code can be degenerate and the code can be universal, and we saw exceptions. So today, continuing that discourse, we are going to be looking at what we call molecular genetic tools. So the idea is that now, what we have done so far is to actually look at observations, what we can actually observe in the natural process. But from now henceforward, we are going to be looking at things that we can do in the lab to manipulate genetic molecules. So if we are going to be able to do that, then we need to know the tools that we are going to use first. Then by the time we know the tools, in the next class, we are going to be looking at the techniques. What are the things we are going to do for the manipulation of genetic material? So today, we are looking at the tools, and the tools will include things like enzymes, classical case restriction enzymes. We will look at things like vectors that will enable us to move genetic material from one host to another host, or from one species to another species. Then we'll look at the expression systems. What are the systems we can use to really grow our genetic molecules or to culture our genetic molecules and stuff like that. Then we'll close the class by looking at what we call the software. Today, there are actually a large number of software that are used for the purpose of studying genetic material. Remember, we are dealing with genetic material that runs in millions and billions of base pairs. So, can you be able to do that manually? The answer is to some extent, but largely no. And that is why software are now on ground that can enable you to achieve some of those things. So we are going to see a number of the software, and then we will close the class. All right. So when we talk about genetic molecules, I mean tools that we were talking about earlier on, we categorize them into these particular classes: the enzymes, restriction enzyme, the vector, the plasmid, the cosmid, up to yeast artificial chromosome. We are going to see that. Then the expression system, typically yeast, bacteria, E. coli, and other ones that we are going to see. Then molecular biology software, vector NTI suite, and many of them that we are going to come across. So when we talk about enzyme, we know that enzymes are actually catalysts for biochemical reaction. They will find enzyme. While we have catalysts that we can use for chemical reaction in the laboratory and stuff like that, we have enzymes that are actually biological catalysts, and they catalyze biochemical reaction within the cell and other entities. Even when we are going to do in vitro studies or experiment, we need enzymes, to apply so that they can catalyze the reaction as we want. them to do because we're applying the appropriate enzyme for that particular reaction. So here you can see nucleases. And what are nucleases? Nucleases are enzymes that will degrade DNA or other genetic material, e.g. RNA, in a stepwise manner. And we can see that there are actually two types. That are the ones that are called exonucleases. That's to say that they deal with this genetic material from the outside part, that's the termini of the molecule. And then there are what we call endonucleases that deal with this molecule where in or within the molecule themselves. So we need to appreciate that. All right. So when we look at the exonucleases, we say that the degrade nucleic acid in a stepwise fashion at the terminal or the terminus of the molecule. An example will include what is listed here: exonuclease 3, BAM31 nucleus, lambda nucleus, and T7 gene 6 exonuclease. So there are quite a large number of them. So we are going to see. So now let's see how this actually. Let's take classical example of exonuclease 3. What does it do? You can see it removes, 5 prime DMP stepwise from the recessive blunt of 3OH terminal. What does that supposed to mean? If you look at this particular diagram here or cartoon, you will appreciate what this particular enzyme does. What the enzyme will do is to degrade the molecule from the 3OH prime towards the 5 prime. The same thing here is doing in this way. And you can see it's breaking the thing piecemeal by piecemeal, piecemeal by piecemeal. Then the other thing we should know about this is the fact that it's non-processive, so all molecules in the population are degraded at the time at the same rate. So you can see what is actually happening bidirectional coming from the right to the left and stuff like that. And then you can also see this one bidirectional, it can eat up all the molecule from all axes and stuff like that. So that is what is called exonuclease. Understand the idea. Exo from the termini, outside. Now outside the molecule. So this is again a classical example of this particular idea in order to buttress the idea of the exo and the endonucleases. So you can see this is your DNA molecule. If you apply exonuclease, it will eat up this molecule from the terminal. If you apply endonucleases, it will go to within the molecule and degrade the molecule. So we'll come to see how that actually works. So in the case of endonucleases, we say they coat this molecule within the molecule or inside the molecule, and classical examples are actually listed. DNS1, RNSH, and RNST1, and the RNSH. RNS1, all are classical examples. So when we take the classical example of RNSH, which is one of the molecules that is actually being used oftentimes in the lab, it cleaves within the RNA strand of an RNA DNA duplex, as you can see here. It's useful for removing the original template strand during a cDNA synthesis. We'll come to see how this is actually very useful and can be used to cleave RNA molecules aside, hybridize to specific deoxyribonucleotides. So remember that we said that this particular enzyme have the capacity to degrade or to break the molecule or to eat the molecule piecemeal within the molecule itself. And that is why they are called endonucleases. So this is your hybrid. The blue color is DNA, the R color is RNA. Remember that you don't have this kind of particular situation naturally happening. It's only under certain conditions where you can have a double-stranded molecule. One strand is actually RNA, another strand is actually DNA. And that is why, naturally speaking, the RNSH exists is in order to take care of this particular scenario. So how does it do that? Let's see how it does that. So here, you actually have in the next step of this reaction, the whole RNA has actually been degraded. You can see piecemeal like that, and that is as a result of the fact that RNSH enzyme is available to do that particular job. Okay, now let's continue looking at the endonucleases. Restriction enzymes. So restriction enzyme can be considered as the antibiotics of the bacterial system. Now, DNA is self-DNA is actually distinguished from foreign DNA by the process of methylation, that's addition of methyl groups. So remember the fact that the bacterial system is a small system, prokaryotic system, and is under constant attack by bacteriophages. So bacteriophages, when they actually attack bacteria, they inject their DNA into the bacteria. So, and then this bacteriophage DNA can go and use the machinery of the bacteria and then develop themselves and then become lytic by killing the bacteria itself. So, nature endowed the bacteria with the facility that they could be able to actually identify foreign nucleic acid, in this instance, the nucleic acid from bacteriophages, so that it can actually take care, because recall the fact that it's a battle or it's a game of survival. All right. Now, restriction enzymes that we call endonucleases, proteins that recognize specific short sequences and code DNA at those sites. You see, they have the facility to be able to recognize certain sequences, of the bases, and then we'll be able to distinguish that, okay, this one, oh, it does not belong here. So, and since it does not belong here, we can actually attack it. And then bacteria contains over 400 of such enzymes that recognize and code over 100 different DNA sequences. So we'll come to see how 400, enzymes are actually recognizing 100 or over 100 sequences. What is that supposed to mean? It means that there is actually an overlap where more than one enzyme will recognize a particular sequence. So let's keep that in our mind. Now, there are actually three major classes. There are what is called restriction enzyme type 1, type 2, type 3. Now let's look at the activities of this enzyme. Under the activities of type 1, you can see they do cleavage and methylation. Under number 2, they do cleavage only, and under number 3, they do cleavage and methylation. Now, so what was that supposed to mean? Is ATP required? ATP is required in type 1 and in type 3. Then cleavage sites, number 1 and number type 1 and type 3, they're actually random. What does random means? Random means they can actually identify certain sequences, but they don't necessarily have to be very specific. Whereas type 2, that is the most popular, that is being used, is the one that will actually recognize within recognition site. That's to say, there are specific sequence of bases that type 2 will actually identify and code. It's not about randomization. No, this one is very specific. You'll be able to actually see it and cut it. For instance, if you have a bunch of keys and you come to a padlock, like I gave a description some time ago, sometimes if you use the wrong key can just talk inside your padlock, and the whole essence of achieving your aim, which is to open the door and enter and do your business, whatever. If it is your house, you want to enter your house and relax and do stuff, or if you are sent to come and pick something within the house, you can enter, pick something and return back. But because you are using the wrong key, the whole project has actually been messed up. So that is the idea here. You have to use the specific key to actually open the lock. And in this instance, you are sure that, okay, this key can always open this particular lock, so that you don't actually mess up the story. Okay, restriction enzyme cutting site actually one specific nucleotide sequence, the DNA at which a particular restriction cuts the DNA. And then some site occur frequently in the molecule, and then every several thousand base pairs, others much less frequently, so rare, e.g. about 10,000. So now see the story. The story is that the longer the sequence of the base pairs, the fewer you actually find them. The shorter the number of bases, the frequent you actually get to find them. And that is what we are explaining in point number 2, that some are more frequent than others. Now, let's see some of the classical situation. under the nomenclature of this enzyme. Let's see the enzyme, the sequences, and the sources where they are obtained. Now we have number one enzyme, we call it enzyme Ig1. The sequence that it will actually identify inside a molecule is what is called GGT. And the corresponding complementary sequence is actually shown there, which is TGGCCA. Where is it obtained from? It's obtained from Agrobacterium genotonovorum. Now, this is how the nomenclature works. You see, if you look at the name of the enzyme, Ig1, you will see that that A is actually brought from the genus name Agrobacterium. And the GE is brought from the genotonovorum, which is the species name. And then 1 is actually the strain. Number 2, we have a DRA1. You can see the sequence. So anywhere this sequence appears in a molecule of DNA, it will actually identify and code it. And then the other one, you can see the source is from the Enococcus radiophilus, and that's where DRA for D, the Enococcus, and then Ra from radiophilus, and 1 strain. PAC, you can see the sequence also. You can see TTAA, TTAA. So what does that supposed to mean? Is 1, 2, 3, 4. That's 8 base pairs. You can see the previous one we have seen there actually 6 base pairs. So what does that supposed to mean? What it means is that the first 2 will be more frequent than the third one, and that one is actually obtained from Pseudomonas alkaligenes. And if you look at the thing very carefully, you will appreciate that it has not actually followed the nomenclature we have stated earlier on, where we said the first letter will be from the genus name, the second 2 letters will be from the species name. In this instance, yes, P is from the genus name Pseudomonas, but instead of al, we are now putting ac. So what is the story? The story is that there is already another enzyme that has already taken that name PAL. So now, since we don't want to confuse it, we need to get another name for this. So that is why you can see it's called PAC1. Then the next is, as you can see here, SNAB1. SNAB1 is from where? It's actually from Spirulithus natans, and this one follows a normal nomenclature. S was for the S, and then Na from the natans, and then B, remember B1 is what? The strain name. So if you identify a particular restriction enzyme from a particular source, this is how to follow to nomenclate it. Remember, we talked about the fact that the restriction enzymes actually cleave DNA as very specific site. We have just seen classical example. Now, when that is actually done, they actually code DNA in 2 fashions. Fashion number 1, creating what we call a sticky end, and fashion number 2, creating what is called a blunt end. Now, we'll see what actually that means subsequently. So your restriction enzyme will recognize and code DNA only of particular sequences. So anywhere, if you have a molecule of DNA that is 1000 base pair length, anywhere any of these sequences appeared, if the appropriate enzyme is actually put, what do you have? You actually get the enzyme coding the DNA at that particular point. So you can see here, Bacterium Haemophilus aegypticus produces an enzyme that is named He3, he from just the name as we have described earlier on, that cause DNA wherever it encounters this particular sequence that is appearing at the bottom here. And what is the sequence? GGCC, CCGG, complementary sequence. So anywhere it actually appears, it actually causes this particular molecule in this particular sequence producing this particular sticky end. All right, so now let's look at these classical examples. Now we have another enzyme that is called HindI3. HindI3 we recognize AAGCTT, and then we'll cut it into what is called a sticky or overhang. This is what is called a sticky or overhang. Then we have another enzyme that we call PBU2. PBU2 will identify CAGCTG. Now, another thing that we have not emphasized in this story is the fact that the sequence that this particular enzyme will recognize is called palindrome. When you take the first strand, you read it from left to right. And then by the time you read the lower strand from right to left, they give you the same sequence. That is the idea of this particular restriction enzyme. So let's keep that one in our minds. We have seen how this molecule can be cut into a sticky end, and we have seen how this molecule can be cut into a blunt end. So classical example of sticky end is actually shown here for us to appreciate. If you look at number 5, I mean, you have these two sources, one from Hindi 2 and then one from another Hindi 3. Now, from different sources, and that's why one is actually red, one is actually blue. So when you apply the same enzyme will recognize the same sequence in the different molecules, cut them into a sticky, and by the time you bring these two molecules together, because of this complementarity bond formation, they will naturally form this bond that you are seeing in number 5, where we say DNA molecule code with the same restriction enzyme have complementary sequence, and that can provide for this particular complementary bonding. But then again, remember there are always gaps. These gaps are supposed to be sealed by what we call DNA ligase. Yeah, yesterday we actually asked question about that. And the reason why we ask question about that is because during the process of DNA replication, we mentioned a number of enzymes, and we also saw the function of DNA ligase. All right, now let's go through this point clearly. If you have a DNA molecule, like we are seeing at the bottom here, and then it's at this length, and then there are actually A, B, C, D, E, and then there are actually 4 restriction sites which are indicated by the red arrows. When we apply our Hindi 4, our Hindi 3 will break our molecule into sticky and in this fashion. So at the end of the day, we have what? We have 5 fragments. 1, 2, 3, 4, 5 fragments. And where are the 5 fragments coming from? Because there are 4 cuts. Now, if we move forward and we want to look into the detail, then we can see another enzyme has been brought on the scene that is called EcoR1. EcoR1 will identify this sequence GAATTC. And by the time we apply EcoR1 is going to cut this molecule for us, giving us a sticky end. And you can see what has actually happened. So when you bring this other half and this other half together, they can always form complementary bonds, and then we'll seal it with our DNA ligase. Now, these are example restriction enzyme and their recognition site: BAMH1, COF1, DRA1, EcoR1, and EcoR2. Their sources where they have been isolated: Escherichia coli, Clostridium formicacetatum, and Bacillus and Mycophacians, and these are the cuts, recognitions are cutting sticky, cutting blunt, sticky, and sticky. And or actually sticky also means cohesive. You understand? So let's know the two terms. You can use the word cohesive or sticky to represent that particular idea. Now, the other story that we need to know about restriction enzyme that actually recognize sequences and code DNA or nucleic acid into pieces is the fact that there are what we call isoschizomas, and isoschizomas are actually enzymes from different sources that recognize the same site. You remember we mentioned the fact that there are about 400 enzymes recognizing over 100 sequences, and I told you it's because there are actually more than one enzyme recognizing the same sequence. So that is where the idea of isoschizoma is coming. Remember iso for the same, schizomas here for cutting. And that is what you are seeing. You can see the same sequence, but cut by 2 different enzymes. Another one, you can see the same sequence called by two different enzymes. So you can actually have some of those kind of classifications like represented in this particular table. Art 1 can actually recognize the sequence, but then it has also isochizomas. Isochizomas echo 147, PME 551, SSAB1, and then STU1. This enzyme will actually recognize that. Then you go to BSUR1, and then you go to the sequence GGCC, and then you can see BISH1, He3, PAL1. So you can see our PAL has actually appeared here, as a isoschizomas to BSUR1. Then we have SMI1. SMI1 will recognize this sequence, and then there is actually an isoschizoma, and the isoschizoma is SOA1. So let's keep this one in our mind. We could actually have different enzymes identifying the same sequence and cutting it. And the term used for that is simply known as isoschizomas. So let us not forget, let's remember that that is the term. And when questions are asked, we should be able to remember and state it very clearly. Okay, let's move ahead. Now we have what we call DNA ligase. DNA ligase, we have just seen, seals the nick that are actually the last thing to be done when tying up a DNA molecule. We have what we call T4 DNA ligase that actually does that work. We also have what we call 4-RNA ligase, also does the same work. Remember, nucleic acid, RNA, DNA. So all these things were sent up RNA and DNA. Let us not forget. They require what? They require an OH, because the OH is the one that will react with the phosphate group to form the bond and stuff like that. So this is a typical scenario. You actually have your molecule here. You have applied restriction enzyme. The restriction enzyme open your molecule forming sticky end, and then this actually hybridize with another molecule. Now, how do you know it's another molecule? Look at the color. The previous molecule is actually blue in color, but we have brought in someone that look like pinkish. So you can see, because due to complementarity, they have actually formed complementary bonds. But then again, you can see the nick, and the nick simply means the gap. If you can easily disassemble this molecule into 4 pieces by just increasing the temperature. Now, in order to put this molecule together quickly, we then apply DNA ligase. The DNA ligase will then seal this nick. And that's why we say DNA ligase seals the gaps. Now, let's move forward to see some of the things. Now, in the case of blunt end ligation, it is actually less efficient. Higher ligase and DNA concentrations are required for you to be able to achieve that. Then the cohesive end ligation is carried between 12 to 15 degrees centigrade to provide for balance between hybridization ends and enzymatic rate. And then blood end ligation is usually carried at room temperature because end hybridization is not required and the enzyme is not particularly stable at temperatures higher than the room temperature. So you also need to take care of that. So you can see why the sticky end is more popular than the blunt end. The sticky end is more popular because it's easier to manipulate, easier to work with, whereas the blunt end will require quite a number of other conditions to be able to work. Now let's see what is happening here. In order to portray this idea, we have also brought in this cartoon for us to appreciate. We have our DNA molecule 1, we have our DNA molecule 2. Our DNA molecule 1 and 2 are actually cut using the same enzyme, EcoR1, in both cultures. And after we have done that, we now brought them together. Remember, they are actually created what we call cohesive ends or sticky ends. And then you can see how we brought them together. After bringing them together, they are able to complement and they have actually formed what we call now, when we say recombinant DNA molecule, what do we supposed to mean? We mean DNA molecule that is actually hybrid, coming from two or more sources. You can see DNA 1 is yellowish, DNA 2 is actually bluish. Then by the time you actually hybridize, you get to see something like this. Molecule 1 can actually have this particular pattern, and molecule 2 can actually have this particular. So this molecule here is what we call recombinant DNA. But you know, after this particular reaction, there is a final reaction, the application of DNA ligase. Don't forget about that. Okay, DNA ligase application, what does it do? We use it to do what we call cloning into vectors. You know, our vector is a molecule that will ferry our molecule gene of interest into the host. So when we are going to actually put our gene, we need to ligate. Then construction of library, you know, DNA libraries, we need to do that. Then linker ligation, we need DNA ligase, and then recircularization of our linear molecule, we also need DNA ligase to be able to achieve that. Next enzyme, DNA polymerase. Now, you recall that what we are doing here is to be able to appreciate what are the things we need to use to be able to manipulate molecules. It's like a chef in his kitchen, you know, wants to produce or want to make jollof rice, you know, what he needs. He needs to know what he needs. He wants to make vegetable soup and pounded yam or gari. He needs to know that, okay, I need gari, I need this, I need to go and buy vegetable, I need to buy guamo, I need to buy meat, I need to buy stockfish, you know, all kinds of things. And then even when he comes to start cooking it, he doesn't put the what do you call the name? Sometimes the oil, sometimes depending on the kind of the oil may be put at the end of cooking the soup. So you don't start doing something earlier that is supposed to be done later. So what we are doing here is that we are trying to understand what are the things we need, you know, when we enter our laboratory and we need to manipulate a particular molecule. Do we need to actually extend the molecule? We need to extend the molecule. That is where DNA polymerase will come in. Do we need to cut the molecule? That is where restriction enzyme will come in. Do we need to ligate the molecule? That is where DNA ligase will come in. So now from our last classes, we have seen the whole story about DNA polymerase. So it's not going to be a new thing that we are going to be talking here. But to just mention the fact that the DNA polymerase from E. coli called DNA polymerase 1 is a classic DNA polymerase. Why? Because it has moderately processive, it's actually a moderately processive polymerase. It has what we call 3 to 5 prime proofreading exonuclease, and it has what is called 5 to 3 prime nick translation exonuclease. Used mostly for labeling DNA molecules by nick translation, and then for other purposes. The clinofragment is usually preferred. And what is the clinofragment? The clinofragment is actually a deconstructed form of the DNA polymerase where the exonuclease proofreading activity have actually been removed. What you need here in the clinofragment is a scenario where you just need the processivity of this particular enzyme. So you have your molecule, the primer is provided, then you can see your DNA polymerase actually extending the molecule. Remember, the molecule goes in the 5 to the 3 prime direction. Always remember that. And that's why in our test of yesterday, we mentioned there was a question. I don't know whether you were able to actually make, since I was not able to review the questions myself to see how the performance is for each of the questions. There was a question that asked that if a molecule is going to be transreplicated or is going to be read, which one is supposed to be, no, sorry, it's been transcribed, which one is supposed to be on top? The one on top, what is it reading? The one at the bottom, what is it reading? The one on top usually is going to be 3 to 5 prime. You get the logic? And then the one that is being transcribed is the one that will go in 5 to 3 prime direction. Now, in this instance that we are seeing on the screen, we have actually flipped the molecule down because here what we are doing, we are actually reading or we're actually extending a reaction. And that's why you can see the molecule on top here is 5 to 3 prime direction. And then the one that is being or that is serving as this template is actually 3 to 5 prime. So let's keep this one in our minds and how to read the molecule. So what are the sources of this DNA polymerase? One, we can have sequences from T7 phage DNA polymerase, highly processed used in DNA sequencing. Then the second one is Taq or Vent polymerase, which is heat-stable polymerases used for making DNA fragments by polymerase chain reaction. Polymerase chain reaction is going to be another topic altogether that we are going to look at. But for now, let's keep in our mind that we need polymerase chain reaction when we want to amplify the number or the quantity of our nucleic acid. Now, Taq or Vent polymerase, Taq polymerase is stable up to 94 degrees and it lacks the 3 to 5 prime exonuclease but not the 5 to 3 prime exonuclease. So, useful for polymerization at high temperature to remove secondary structures and for thermocycline reaction, e.g. the polymerase chain reaction that we're talking about. And you know, the discovery of this particular idea was just by serendipity. But for now, let's keep quiet. When we get there, we can explain. Now, Taq polymerases that are used in this particular PCR reaction, as like other enzymes, are actually sold in things like this small ampoule, very small plastic bottles like this, micro tubes like this, and these micro tubes, as you can see, very expensive. I use these to actually describe what we call the wealth of nations. Now, you see, some countries have gold, Burkina Faso, for instance. In fact, just this afternoon, I was watching a video on YouTube and they were saying that Burkina Faso has no, millions of tons of gold, you know. Then when you leave Burkina Faso, you go to Niger, God has blessed them with uranium. When you leave Niger, you come to Nigeria, God has blessed us with oil. You know, so everybody has his own blessing. But some people don't have those blessings. What God has blessed them with is their brains. So they are using their brains to enrich themselves. And how are they doing it? Let's look at a classical example. You see, this is a Taq polymerase from New England Biolabs. And if you look at this thing, it's a very small tube. And this tube can come in three sizes, small, large, and extra large. And when you see extra large, it doesn't mean anything. Oh, it's just something small that you can actually put in your mouth, and nobody will know that you have put something in your mouth. And how much is it costing? It's costing almost 600 US dollars. Now, see the issue. If you produce one tube of this particular enzyme, costing almost $600. Remember, this is equivalent to like 7 to 8 barrels of oil. My friends, there is no way you can, no matter how you try to conceal, there is no way you can conceal 8 barrels of crude oil. How are you going to carry it, by the way? Are you going to carry it on your head? You need a truck to be able to carry that. And this is something equivalent to just a small tube that somebody has put in your mouth and will come and pass in front of you without you knowing. So now the question is that who is richer? Anyway, let's move forward. Tag polymerase, you know, they can come in this now polymerase tag, PFU, and vent. Exonucleotide 3 prime to 5 prime exonuclease, no for tag, yes for P, and then for vent, yes. Source and properties are stated there from Thymus aquaticus. Half-life is 95%, 1.6 hours. And then for PFU, you actually have from Pyrococcus furiosus, appears to have the lowest error rate for non-thermophilic DNA polymerases, and for the vent from Thymococcus littoralis, also known as TB polymerase. Half-life is actually 7 hours. So these are the now. Why will you want to choose any of this? You want to choose any of this because you want to choose the one that will service your purpose. Just like you, when you think you have money, you want to go and buy a car for your family, you have to sit down and plan. Oh, this car I want to buy, what is going to be the use? Is it for me to go to office or is it for me to be traveling with my family? And we need to have convenience, we need to have comfort. So what kind of car will be the best? Or is it that I want to be going to farm? If I want to go to farm or I want to go to the field for some, if I'm an engineer who is into construction and stuff like that, my friend, it's not every kind of vehicle that will serve all kinds of purposes. Especially if you are a farmer, you cannot go and buy a luxury car and be taking it to the farm. So that's the same thing here. You have to decide which of these enzymes will service you the best for understanding the experiment you are actually going to be running. Now we are finished with DNA. Let's move to RNA. Because again, don't forget we are going to be manipulating RNA molecules. And if we are going to be manipulating RNA molecules, sometimes we may need to extend an RNA molecule. And you can see here we have what we call RNA polymerase. Now RNA polymerase from where? From DNA. Now see, usually from bacteriophage SP6, T7 or T3 and can be used to generate large amounts of RNA complementary to sequences downstream of the appropriate bacteriophage promoter. And then the best method for producing large amount of homogeneous RNA for use and hybridization probe. T7 application, what I would do, we use it for radiolabeling RNA probe preparation, we use it for generation in vitro translation, we use it for generation in the studies of RNA, structures processing and catalysis, and then we also use it for expression control via antisense RNA. So these are some of the applications that tRNA is actually used. Remember, you want to produce RNA from where? From DNA. If you are going to do that, then you need an RNA polymerase because that is the enzyme that polymerizes RNA and one of the sources T7. The other one is what we call phosphomonoesterases. Now, what does phosphomonoesterases do now? Reverse action of polynucleotide kinase. Now, polynucleotide kinase enable us to add phosphate to a molecule or to a biological structure, whether it's protein, whether it's whatever. Now, you see, this phosphomonoesterases, what it does is to remove that phosphate from, remember that, phosphorylation of an organic material is actually energization of that particular material. So here, what phosphomonoesterases will do is to remove this phosphate, and that's why we say it's actually a reverse. Where do we get it? Bacterial alkaline phosphatase is a classical and traditional source. And then we also have what we call calf intestinal phosphatase. It's newer, easily inactivated, and is preferred. So what reaction do they actually catalyze? The reactions are actually shown here. In the case of DNA, when you actually carry your DNA molecule or solution, you apply alkaline phosphate. What they will do, what alkaline phosphate will do is to go and remove the 5' phosphate. And then you end up with a molecule that doesn't have a phosphate group. What does that supposed to mean? It means that this molecule cannot be extended, whether at each other point, because there's no what. Then the other thing you can do with phosphomonoesterases is to actually remove completely, including the ones that are inside the neck. You can see here under standard condition, you remove this other one, but the one that is within the neck is still intact. But under slightly denaturing condition, if you look at it, the phosphate that is here have also been removed. So what do? We remove 5' to 3' phospholipid, groups from nucleic acid. Number two, preparing templates for 5' labeling. Number three, preventing fragment from cell ligation. Number four, dephosphorylation of protein. We want to dephosphorylate protein, we use phosphomonoesterases. Now let's go back to point number two. We want to do template 5' labeling. What does that supposed to mean? We want to apply isotopic phosphate. Application of isotopic phosphate will enable us to produce a probe, that can be used or that can be identified. Okay, let's move forward. The other one, RNA dependent DNA polymerase is actually a reverse transcriptase. Remember in our lecture on central dogma molecular biology, we say that the RNA can actually be reverse transcribed into DNA. What does that? What does that is this particular enzyme known as reverse transcriptase. And as you can see, when that is actually done, the next thing we need to also do is to apply our RNSH. Our RNSH will actually remove the hybrid, that is the RNA from the molecule, and then that molecule can then be now used as what as a template to produce a second strand. So this is what you are seeing here. You have your RNA molecule, you want to reverse transcribe, and that is why the name of the enzyme is RNA dependent, because it's depending on the information provided by this RNA molecule. Then from there, you provide a primer, and then from there, you can see your reaction after the provision of the DNTPs, DHTP, DGTP, GCTP, and stuff like that. And then with the enzyme, you can see the DNA molecule strand has actually been formed. So this is the molecule that RNSH needed to remove this other strand of RNA. So um, this reaction is shown here for us to really appreciate. RNA molecule is red. The primer has been attached. We provided all the condiments required, the ingredients, the DNTPs, the enzyme. So the molecule has been produced. When this molecule is produced, remember this molecule is not supposed to exist in nature. And because of that, the RNSH is actually here. Or we can also apply alkali, any alkali that can do the job. After that, you can see your molecule is actually removed. And now you have your single-stranded DNA molecule. Remember again, single-strand DNA molecule is not naturally existing. And that is why DNA polymerase will now be applied. And this DNA polymerase that is applied in the presence of what we call the DNTPs would then generate and produce the other strand. Now, but you see where there is a problem, there is a failure at the terminal here. A loop, for whatever reason, a loop is actually formed. This loop is not part of this particular molecule because DNA does not exist in single strand form. So that is why another enzyme that is called single strand specific nuclease will then be applied. When it is applied, it will go and remove this looping and remove it out. Then you now have a normal DNA molecule. Now remember our equation, DNA to RNA, RNA to protein, and then you can see the reverse version. So this is where we have just talked about, you know. So DNA dependent RNA polymerase and then RNA dependent DNA polymerase. So our reverse transcription reaction and stuff like that. So where can we get this? We can get this from murine leukemia virus, the reverse transcriptase. And then what will they do? Remember we said we can produce our cDNA preparation both first and second strand. And then number two, we can use it for DNA sequencing. Then the other enzyme that we need to also talk about is called terminus transferase. When we say terminus transferase, what does that supposed to mean? Like the name implies, it's going to be adding, nucleotide for us at the terminus of the molecules. Where do we get them from? We get them from calf timers, and it adds DNMPs, from DNTPs onto the three prime ends of DNA chain. In this instance, acceptor as short as 3 nucleotides can be used. Is stimulated by cobalt and then useful for adding homopolymeric tails or single nucleotide can be labeled to the 3 end of DNA strand. So let's see what we mean by that. Now, look at it. We have our molecule. Can you see it? It's actually a cohesive molecule on this side and on this other side. Then we apply our terminal transferase. In this instance, we can determine very specific terminal transferase. And the reason is this. We want to identify it so that we can be able to tell with precision that, okay, This is what we want to achieve. So you actually add T polymeric T, and then at this point you can actually bring your molecule. And your molecule, depending, since you have added T here, the best for this is actually going to be A. Then you also have this one, your molecule is linearized, and then you bring in your what? Your terminal transferase. And since your molecule is already poly G, what is the best? The best is going to be poly C. And so you prime this one with terminal transferase using the DCTP. At the end of the day, this your fragment is going to be inserted by way of complementarity, and then you have this portion to cover. So you can see how terminal transferase can be used to actually produce a circularized molecule. So what are the application of terminal transferase? Now, before we talk about the application, the other thing I want to emphasize here is that terminal transferase does not require a template. Remember that DNA polymerase requires a template. So addition of polypol or homopolymer tails to the 3' ends of DNA is one. Number two, labeling the 3' ends of DNA. Now, because you are the one that is actually trying to achieve this particular purpose. The next enzyme is what is called T4 polynucleotide kinase. Earlier, we have actually seen phosphomonoesterase that removes, phosphate from the DNA and from proteins and stuff like that. Here, we are seeing another enzyme that adds phosphate. That's why it's called kinase. Anywhere you hear the word kinase for an enzyme, it means it's phosphorylation that is taking place. So you obtain it from T polynucleotide kinase, which transfers the gamma phosphate from ATP to your 5' OH end of the molecule. Remember that at the 5' there's always a phosphate, but you have used phosphomonoesterase to remove it. Now you want to return it, so you use polynucleotide kinase to do that. Now, if the gamma 32 ATP is used for substrate, the nucleic acid recipient becomes radioactively labeled, which you can do. This is probably the most commonly used method for N-labeling of nucleic acid. Let's see what actually happened. This is our molecule. We have already removed the P, the phosphate using phosphomonoesterases, and then we now want to return it, so we can do and then provide, and then you can see our gamma phosphate is transferred from ribosomal R ATP. Now, the other reaction that we need to show here is that in a full reaction, this is what actually happens. In the presence of polynucleotide kinase ATP, we can actually provide our phosphate. And this order is actually an exchange that is taking place. This is the normal. So by the time we apply it, we can actually remove it, and then we bring the isotopic phosphate, and then we can actually replace. So this is actually an exchange that is taking place. You remove the normal through this reaction and then add. Now, what makes the difference? What makes the difference is that in the presence of ADP, this particular P is removed and added, and an ATP is formed. Then, in the presence of this particular ATP again, the polynuclear kinase can actually phosphorylate this molecule by returning the phosphate, as you can see here. And that is what is called exchange reaction. So polynucleotide kinase will enable us to add, phosphate to the molecule. So unlabeled ATP can be used to convert 5' OH, which we have just seen. Then recessed 5' are not good substrate. Then unusually sensitive to contaminants. It requires highly purified nucleic acid for this particular reaction to actually occur. Applications are N-labeling, we have just seen. Addition of 5' phosphate to nucleotide, we have just seen from the equation. And then removal of 3' phosphoryl groups, again, is another way of actually dealing with that. All right. That's just a summary of few of the enzymes that you may frequently encounter when you are actually doing molecular studies or experiment in the lab. There are many enzymes. There are many enzymes. We are just in some of the few basic enzymes. All right, now let's see how about the vectors. The vectors, we say they are the ones that enable us to actually transfer our molecule of interest into the host. So we are going to see how this particular vector does that. But then we can say we need them because they will enable us to also replicate our molecule and stuff like that in the host. In 1973, from our history, we saw this particular group of people who have actually contributed to our understanding of this particular entity called the vectors. Stanley Cohen and Annie Chen and Herbert Boyer. These are all the people who have pioneered ideas into this. So you can see they use it to actually transform E. coli with a recombinant plasmid. How are they able to do it? They got their plasmid. This plasmid is called PSC101. P standing for plasmid, S standing for Stanley, C standing for Cohen, 101 basic plasmid. They were able to use restoration enzyme to open it up and then brought in their gene of interest, in this instance, kanamycin resistant gene, and inserted it here. And then they use it to transform E. coli. Remember, transformation is the process of picking DNA molecule or material from the environment by bacteria. And because this plasmid has a kanamycin resistant gene, by the time they put it inside a culture and apply kanamycin, the cells that were able to pick up the DNA were able to survive because the gene was able to express within the bacterial cell that picked it up, and then it conferred resistance to them to kanamycin. Now, vectors, especially plasmid vectors, are circular in nature, as you can, and many other types of vectors. One of the things we must keep in our mind is to be able to appreciate what are the common properties that actually describe a vector as good. Number one, it must have a cloning site. That the cloning site is the site that you can use to cut with restriction enzyme so that you can introduce your gene of interest or DNA of interest or insert. Now let's see, this is our vector, PST1, Echo R1, CLA1, IND3, BAM-H1, SAL1. These are classical restriction or cloning site. Number two, a replicator. What is replicator? Is replication a replicon or origin of replication? We can see that this blue segment here is the origin of replication. That means that this molecule can replicate. Then number three, they must have a selectable marker. And what are the selectable markers here? The selectable markers here are the ampicillin resistance and the tetracycline resistant genes. You can see them here. So for instance, if you intended to use tetracycline in your experiment, then you must be sure not to use cloning site within this region. You cannot use IND3, you cannot use BAM-H1, you cannot use SAL1. But you can use PST1 because PST1 is in the ampicillin resistance. But if you are going to use ampicillin, then you cannot use PST1, but you can use any because they are outside the resistant gene. So these three ideas are very useful for a good vector to actually have. There are quite a number of types of vectors, and they are listed here for you to appreciate. You have the plasmid, you have the cosmid, the lambda, the phage, the phagemid, the plasmid. And then you have what we call shuttle vectors. We are going to see what shuttle vectors. We have the bacterial artificial chromosomes. We have the yeast artificial chromosome. We have the one specific for plant that are called TI plasmids. And then we have eukaryotic viruses such as baculoviruses, lentiviruses, you know, And here you have SV40 virus and some other retroviruses. Now, classical example, remember we mentioned the fact that these vectors are usually circular, so you can see a classical case here. BACPA-K6, the size 137 kb, and then you can see the open reading frame. You can see the cloning site, BSU361, BSU361, BSU361. Now, this table here describes or gives you a general characteristics. You have your vector, plasmid down to YAC at the artificial chromosome. You have the host where you can actually clone them. You then have the carrying capacity, how much quantity of DNA they can carry. You know, for instance, plasmid can carry up to 15 kb. If you have 32 kb fragment, that means plasmid cannot do that job. So you need to go up to look for the appropriate. In this instance, your 32 kb will be appropriately carried by a cosmate. Now, if you have 172, that means you may likely use bacterial artificial chromosome. And then if you have a 452, then you know that you cannot. Yes, what you can use a bacterial artificial chromosome. There's no big deal about it. But even if you have your yak, you can still use it. And then the major uses for that is that. Now, let's give a caution here. Why this table is important is because you should be able to keep in your mind the carrying capacities. You cannot carry, you cannot use a vector that the molecule you have, the carrying capacity is higher than that. But you can use one that the carrying capacity is higher without much problem if you can manage and handle them. So, PBR 322 vector is one of the most common plasmid vector that is used. So the size is there, 4.36 kb, and all the restriction sites are actually shown. The origin of replication is shown. The resistant genes are actually shown, which are ampicillin and tetracycline. You know, and then the cloning side, they are all actually shown here for you to appreciate. Then, like I said, the one that is used for plants is actually called TI plasmid. This TI plasmid is actually came from what we call Agrobacterium tumefaciens, which contains plasmid, and they are actually ranging within this 100 to 200 kb, which the tumor-inducing genes are actually located as the TI plasmid. Then the part of the plasmid that, excuse me, the part of the TI plasmid that is transferred to the plant genome is what is called the tDNA. So the tDNA actually has the virulence genes as genes and then the open catabolism genes. Now let's see what that means. So the tDNA contains genes essential for synthesis of open, and the open produced by the agrobacterium determines the name of the TI plasmid it carries, e.g. nopalin, octopine plasmid, and stuff like that. So if you have seen a plant that has formed callus, like you can see in this particular picture here, actually shows that this particular callus has actually been orchestrated by the presence of TI plasmid. So this is the gene, and in this gene, sorry, this is the plasmid, and in this plasmid you can see several segments. You can see origin of replication. You can see open catabolism, and then the tDNA region, you can see it here, from the right border to open to cytokinin to auxin, and then to the left border. Now, remember we said this is actually what enables this virulence to actually occur, e.g. the formation of this tumor you are seeing on this particular plant. So, the T plasmid can be used to transfer gene into plants. That is why, oh, no, let me not say all, but essentially, the TI plasmid is the most popular means by which DNA is vectored into plants. And a typical example is actually captured for you in this particular cartoon. You have your plant growing, And then Agrobacterium tumefaciens in the soil can actually affect the root. And then this particular root can actually be bacteria can actually go into the cell. And you see the cell here, and this is what is called the infection process. The 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 plasmid from the T, I mean from the T plasmid from the bacteria is actually moved into the cell and eventually enters into the nucleus of the plant cell and gets integrated there into one of the plant chromosomes. And then from there, it can continue to multiply itself. Then number B, for instance, here you can see your molecule. This is your selectable marker. This is your foreign DNA that you want to actually move. You bring them together, select everything, and then you have your foreign DNA inserted into this particular plasmid, and then you can see what actually happens. This cell or the bacterial cell has picked up this particular plasmid, you know, and then you can see through the same process that is used for a cDNA plasmid or the PI plasmid is actually used to move into the plant cell. So that is how you actually have your molecule transferred into a plant through this particular process using the TI plasmid. The other vector, which we quickly mention, is what is called the cosmic vector. The cosmic vector actually features lambda and plasmid. That's why the name is called cosmid. Like other plasmid vector, but also contain a cosite, naturally occurring cosite, that's cohesive site. So usually they are used to clone larger fragments, approximately 40 kb. So this is a typical scenario, where you have your original replication and then you have your core sites. And you see them here. And then you have collection of multiple cloning sites. In this multiple cloning site, these are the cloning site, Echo R1 Node 1. And then you have BAM H1, you have Node 1, you also have Echo R1 at the end. So this is actually a mirror image from this other side coming this way and from this other side going up. Now, the size of this particular Super Cos 1 Cosmic is 7.6 kb. So, remember that actually cosmic vectors do not exist naturally. They are a product of laboratory manipulation. So what actually happens is that we are getting plasmid portion and then bacteriophage lambda and bringing them together to produce what is called cosmid. Now, mid from plasmid and then the cause coming from the cohesive end that actually naturally exists in phage lambda phage. Now, you can see this is the molecule of phage lambda. You can see the cosite. You can see the cosite. This is your genomic DNA that you want to actually transfer. And then this is your plasmid. Can you see? Your plasmid has its own cosite, this cosite, and then you have your genomic DNA on this side. So you now bring your lambda and everything, put them together, and then you can see ligation. So you can see that your molecule from your your cosmic vector actually has integrated into this. You can see the color representing, and you can see your tetracycline. So you can see your coside, 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 and coside. So this is what is actually used to be packaged into bacteriophages, and these bacteriophages will eventually infect a bacteria. And because what they do after infecting bacteria is transfer their genetic material into the bacteria. So you are using them as a means by which you can cargo your genetic material into the bacteria. So that is the beautiful use for this particular vector. So once they are inside the cell, DNA circularizes by annealing to its positive ends and then replicates as a large plasmid. Then packaging and infection by fetching lambda in extract makes plasmid nearly as efficient cloning vector as lambda itself. Now remember, because the bacteriophages had the power to send in there, so that is what we are actually trying to use. Remember that with that, we can actually transfer up to 40 kBA of our insert. Then the next is what is called bacterial artificial chromosome. It's actually based on E. coli fertility and then copy number 1 to 2 per cell. And they actually accept larger fragments up to 300 kb, transfer into E. coli by electroporation or electroshock, and then changes permeability of the membrane. That is why you are able to do that. It provides low yield of DNA, is stable, and do not tend to rearrange. That's the beauty of the reaction. So your bags are helpful for complete coverage of large genomes. The vector requires E. coli, F factor, replicon, selectable marker, and cloning site. Remember, you have to get all this in on your vector, because that's what makes a vector a suitable or good vector. And then insert up to several hundreds will be taken, low copy number per cell, relatively stable, resistant to rearrangement. That is the beauty part of this particular, and that is what you are actually seeing here. You can see a bacterial plasmid. Then you can see on this side your gene of interest. You have cut it out. You have brought it together into this particular. But so you can now see that essentially this molecule is actually made up of your gene of interest or your DNA insert. You can see the larger portion and the small portion of your what? Of your bacterial plasmid. And then with this, when you come, this bacteria will not be able to pick up this naturally because of the size. So that is where the idea of the bacteria here is electroporated into. What does electroporation mean? Electroporation means manipulating the culture in a way that you send in light or weak electric signal into the culture so that it will ginger the cell membrane and the cell wall for the for the openings in the cell membrane to actually increase in size so that they'll be able to pick up this your material from the environment. And then you can then select it. And that is the selection that you actually see in this column to show you which ones have actually picked up your DNA from the environment. When they do, they actually produce this S-gal color of blue. So lastly, the yeast artificial chromosome is very helpful for complete coverage of large genomes. The vector requires yeast origin, yeast selectable marker, yeast centromere, yeast telomeres, and cloning site. Can take up more than 1 million base pairs. Low copy number per cell. Then possibility of rearrangement during cloning or propagation actually occurs. So this is a typical scenario of your yeast artificial chromosome. There are telomeres usually at the end. Remember that telomeres are characteristics of eukaryotic chromosomes. And then there is origin of replication, and then there is the centromere, the point at which two chromatids can actually attach. And then there is the multiple cloning site, and then there is the sequence for selectable marker. Remember that you will need to select cells that have been transformed from the ones that have not been transformed. Then you have your yeast artificial chromosome, and then you have your DNA from human source or from other mammalian sources. Remember, very large genes, that can go up to 800, 500, 800, even up to 1 million base pairs can be carried by this particular. So what do? You bring your yeast artificial chromosome, you open it up, with a particular enzyme, in this instance BAMH1, and EcoR1 digestion can actually take place, and then you open up your molecule like this. When your molecule is open up like this, you then bring in your DNA from human that have already been caught using EcoR1 digestion, and then you put them in the same environment. By the time you do that, you can see DNA and yeast cell transformation has taken place, and because that has taken place, what do you have? You have this portion of your yeast artificial chromosome, and then this other portion of this artificial chromosome. Now, where is your DNA from the human source? Look at it here. Where is it inserted? It's inserted in what is called the multiple cloning site. That is where you actually have your open restriction. So you can see up to 1 million base pairs have actually been taken up. Remember the reason for considering the different types of vectors. The idea is for us to ascertain what we can actually do. We have a small gene. The gene is 7 kb. What do? We go for a plasmid. We have a DNA insert of 33 kb. What do? We go for a cosmet. We have another one is 132. What do? We go for bacterial artificial chromosome. Oh no, we have another one that is 632 kb. What do? We go for a yeast artificial chromosome. So keep that in your mind. Now, looking at that table moving forward is what we call shuttle vectors. What are shuttle vectors? Shuttle vectors are vectors that will enable you to shuttle between prokaryotic system and eukaryotic system. In this instance, from bacterial system to yeast system, or what we call Saccharomyces cerevisiae, very important. So here is a typical diagram to illustrate this idea. So if you look at this, you will appreciate something. This is your shuttle vector. In your shuttle vector, you have original replication for bacteria. You also have original replication for yeast. These are very important ideas because the prokaryotic system will not be able to deal with the eukaryotic original replication. So, and since you are going to be moving them from one place to the other, then your multiple cloning site is up there, your resistance for gene is here, and then using 2 genes for selection yeast is also provided. And so you can see under antibiotic resistant bacterial transformants actually here, and then under leucine 2 yeast transformers are actually seen here. So this is what is called a shuttle vector. The shuttle vector to the shuttle between eukaryotic and prokaryotic system. So that ends the story of vectors. Oh, sorry, this is the last part, which we call expression vector. Expression vectors actually have the transcription and translation signal necessary for the expression of that particular gene. This is a classical case. Now, remember, we have our cDNA. Our cDNA is actually our gene that we have already removed the introns, and we have converted it back to DNA. So it becomes our complementary or copied DNA. This complementary or copied DNA, we can then put it in our expression vector. If we do that, what we want to achieve is that we want to produce this protein or polypeptide inside our host. And that's what has happened here. After doing that, we use it to transform this bacterial cell, and the bacterial cell is now producing the protein for us. This protein can be e.g. insulin. This is one of the ways by which they actually are able to produce insulin in commercial quantity, through this particular process. The nesting expression system, usually living host, whether they are bacteria, in this instance E. coli, whether they are eukaryotic in this instance Saccharomyces yeast, Cerevisiae, Pistia pastoris, or whether they are viruses, baculoviruses, adenoviruses, lentiviruses, as I mentioned earlier on, and then mammalian tissue culture, and then transgenic animals, e.g. flies, mice, and barnyard animals. So a typical example of the pictures are actually showing multiple host systems are commonly used to produce recombinant human proteins, just like we have seen in our last expression vector. So E. coli, yeast, insect, mammalian cell can actually be used. Different hosts for different products. E. coli, Pistia pastoris, Bacillus subtilis, and you can see what they do. If you take E. coli, for instance, expresses greater than 80% of commercial microbial products, expresses simple to moderately complex proteins, including carbs. Soluble expression simplifies recovery and downstream processing, produces aglycosylated proteins, and ideal for plasmid DNA production. That's E. coli. In the case of Pressure pastoris, you can see expresses larger complex protein and small polypeptides. Solubility issues in E. coli may resolve with picture, and then secretes for easier recovery and downstream processing, and then lacks endotoxins. So then by the time you go to Bacillus subtilis, expresses simple monomeric proteins, soluble expression, amplifies recovery and downstream processing, lacks endotoxin, and then produces aglycosylated proteins. So these are the things. Remember, we say different hosts for different products. So it all depends on what you want to achieve for you to select. Now, expression system can also be specialized. In this instance, you can have Xenopus labus oocyte and then in vitro transcription translation. This is actually happening right in the class without actually the use of a vector. So what enables you to choose the best? Number one, yield. Number two, product purification. Number three, post-translation. Is there a post-translation modification required? Number four, genetic manipulation of the gene or cDNA. Number five, the cost. And then experimental difficulty. Do you have the capacity to handle it? If you don't have, you need to go and get that done somewhere for you. Bacteria is very direct. E. coli, for instance, very direct. And why is it very direct? Very direct because for years, scientists have been studying E. coli and they have understood the basic biology and physiology of the E. coli. So number one, it actually has simple physiology. Number two, short generation time as bacteria grows and multiply rapidly. Number three, large yield product, which is very important. Okay, 10% of the mass, and then at a low cost. So it becomes very important. But then it has some disadvantages. And what are the disadvantages? Number one, express protein often do not fall properly. Remember that you want to produce or express eukaryotic protein inside bacteria. Bacteria don't have the complete machinery to do that. And then synthetic protein is often toxic to the bacteria. Again, that is another problem that needs to be taken care of. Now, they also lack enzyme response for post-translational modification. Oftentimes, the complex protein will need to be modified after production. So this machinery does not actually exist in the bacteria. So that will also become a problem. Then the other set is actually the yeast. Yes, it's actually simple. The eukaryotic, but the advantages are the fact that yeast performs many of the post-translation modifications that are required for a human protein system and then can be induced to secrete certain proteins into the growth media for harvesting. E.g., hepatitis B vaccine, for instance, has actually been used. But we have disadvantages. It will naturally, there are presence of inactive proteases that will degrade the foreign protein is there. And so we will need to find some solution to that. Then the next is the insect cell culture that can be used to express this as a vector, as a host system. So foreign protein in insects through in insect cells through incorporation of their genes in bacillus viruses can actually take place. What are the advantage? High level of expression, correct folding, because now this system actually has the machineries that are needed to perform this function. Post-translational modification similar to those in the mammalian cell, but then cost, though more than that of culturing bacteria and yeast, but less of that of mammal. Yeah, so you can appreciate. And so potential vaccines for HIV virus produced by expression in this system have been happening. And there are disadvantages, more difficult to work with. Yes, because the system is becoming more complex. Then you have it's expensive. Generation time is slow because it will take some time, for the cell to actually uh, develop, grow and divide. Then not suitable for proteins with repetitive sequence. So these are the disadvantages you get to see with the insect cell culture. About the mammalian system, which is their ultimate target, because yes, why we want to treat all animals, we want to relieve animals of their disease, but our major aim is to produce, solutions that will be directly applicable to man. And man belongs to this particular system that is called the mammalian system. So the eukaryotic expression system may be necessary to produce some types of proteins, such as protein that will require post-translation modification and properly folding or proper folding is very important. So the system, many types of mammalian proteins actually there to produce this particular. Then study protein function, very important in their native environment, then produce biologically active molecules, and then introduce new genes or alter existing ones in the intact animal. These are all part of the way that is used to actually study and understand the phenomena surrounding certain physiological processes. Now, two ways that we can actually transform a cell. There are what is called transient transfection of mammalian cell, and there's what is called stable transfection of mammalian cell. We are going to see that subsequently. In the transient, which means temporary, so you get your cell, the defective HB40 provide active antigen gene, then you use it to transfer the cell. You can see your expression vector has actually moved into the nucleus. And then from there, you can appreciate what happens after 48 hours. It has replicated, and then the ribosomal, the messenger RNA has been produced and has been sent into the cytoplasm, and then you can see the machinery that is actually taking place, the activity is actually taking place. Yeah, protein systems complex, and all these blue dots are actually produced, or the protein that have actually been produced. Now, remember, they are standing by themselves inside the nucleus. They do not integrate into the host genome. That's the difference. Now, let's move to the next. In the next, what you see happening in what we call stable transfection, what you see happening is that after the vector enters into the nucleus, it finds its way and integrates into the genome of the host. That's why you can see this black side here and this black side here, they represent the genome of the host. So this will be expressed anytime the genome is being expressed. It will go to the cytoplasm and it will produce. Now, what is the difference? The difference is that this one is now part and parcel of the cell. That is why it's called stable transfection. So if you want to produce this particular protein over and over and over for a long time, this is the pathway you should follow. You produce what we call stable transfection. But if you just want to do temporary, then you do what we call the transient. So what are the mammalian system advantages and disadvantages? Number one, it is used for cell biology, localization, cell cycle studies. Then two, post-translation modification. Then three, improving expression vectors and transfection methods. These are all the strategies we can actually understand and then apply. Are there disadvantages? Why not? Number one, low yield of expressed proteins. Number two, relatively expensive tissue culture course and the rest. Number three, mostly laptop. Remember the shuttle lecture stop is the fact that you can move it from prokaryotic system to eukaryotic system and vice versa. So these are the ideas we have just mentioned about the transient and the stable transfection, which you must remember. One is temporary and then one is actually some permanent or stable. So as we close this particular class today, I would like you to really appreciate the idea that today in biology, software is heavily used. Some years ago, before the biotechnology revolution and the molecular genetics revolution, people didn't think that biology would need to use software. They only thought that, okay, maybe physics, maybe computer science, naturally, they are the owners, and then physical sciences, engineering, but they never thought that people in biology, people in genetics, people in biotechnology can actually use software. But today we can see it has become a common tool. So some biotech software are actually listed here: BioNavigator, BioTools, Genesis Enterprise, Imaging, Ingenuity Hardware, OligoMaster, Silicon Genetics, Vector NIT Suite, Virtual Chemistry. This enables you to do different kinds of things. You want to create oligonucleotide, then you need to get things like OligoMaster, because that will enable you. You want to develop network, of proteins and stuff like that. You need Ingenuity Pathway analysis for you to be able to appreciate. And then you want to actually simulate reaction, you need virtual chemistry. Some of the producers include BioDiscovery, Informax, Ingenuity System, GeneCodes, Unleashed Informatics, Premier Biosoft. And now, right now, we have big companies like Google actually deep into so many of these systems, including their subsidiary called DeepMind, that is the model creator for what we call AlphaGo. AlphaGo, which is actually the people who produce, the DeepMind, the people who produce what we call the AlphaFold. The AlphaFold is a platform that actually have decipher the folding of millions of proteins. So today with that, we are understanding how proteins actually work. It's not a small feat, and because of that, the DeepMind got the Nobel Prize for chemistry last year, 2024. It's a big feat. So these are collections of proteins and software that you can actually use, to perform quite a number of genetic analysis, whether you are dealing with DNA, protein, or all kinds of things. Now, recall we talked about plasmid. This is actually what we call plasmid-32. Plasmid-32 is essential for enabling you to draw the structure of plasmid, as you can see here. Let's take some time to look at this particular window of plasmid-32. There is a plasmid here, and it's called YEP. This is yeast plasmid. So you can see here. Can you see the original replication region? You can see the original replication. Can you see the resistance? Ampicillin and tetracycline. And can you see the cloning sites all over the place? So this particular software will enable you to draw up your plasmid or your vector very quickly. Now, there's another one here that is called STRAP. STRAP standing for structure or sequence alignment program STRAP. Now, what you do here, you actually try to look at sequence of proteins, amino acid on a polypeptide. You pick on a particular polypeptide, in this instance from Saccharomyces yeast, from Xenopus laevis, from humans, from rats, from mouse, from Drosophila, from the clan Arabdosis thaliana, from the worm Caenorhabditis elegans, and you try to relate now, it will align this thing for you, and then you are able to do sequence similarity studies, so that you are able to see how similar are the amino acids that you have picked up in of a particular enzyme, for instance. You understand? Let's say fructokinase. If there is our enzyme fructokinase, we look at the sequence one by one. Then we are able to establish how far a particular species from, because the ones that are close, what that means is that the similarity of their amino acid sequence is very high. You can get up to like 80%, 90%. If you compare to another species and you are getting 50%, it means that those two species are actually far apart. All right. Now let's.
With SnapGene, you can create and annotate sequences, design primers, and plan, simulate, and validate cloning experiments. SnapGene can also align DNA or RNA sequences and simulate agarose gel electrophoresis. Let's demonstrate. You can define a folder as a project to manage and work with your files directly within SnapGene. Let's import a raw vector sequence into SnapGene. Paste the sequence into the New Sequence window, and SnapGene will automatically annotate the sequence using a curated library of common plasmid features. And just like that, we've created an annotated plasmid displayed as a tab in the main SnapGene window. Save the new sequence to your project folder. This map view can be changed using the left toolbar. For example, we can show or hide restriction enzyme sites, find predicted open reading frames, and change how features are displayed. To view the sequence, switch to the sequence view. Here you can also control the view using the left toolbar or type directly in the sequence view to edit. SnapGene will automatically translate coding sequence features. If no stop codon is present, the translation will continue until the next stop codon. This makes it easy to spot errors and understand in-frame fusions. This new sequence was annotated using SnapGene's built-in library of common plasmid features. However, you can also save your own custom features and then detect them on your new sequences. You can easily choose what enzymes to display on a sequence and save custom enzyme sets. There is also the ability to simulate agarose gels in SnapGene. Assign different sequences to different lanes and choose different combinations of restriction enzymes. SnapGene makes it easy to work with primers too. If you have existing primers, store these in a delimited list and import matching primers onto your sequences. You can view the primers on the sequence and as a list in the primers view. It's also possible to manually create your own primers. Just highlight a region of the sequence, confirm the TM, and add the primer. You can also add extensions to the primers, such as restriction sites or nucleotides. SnapGene will automatically design appropriate primers for PCR in cloning experiments, and assist with creating complex assemblies using one or many inserts. SnapGene can simulate PCR, restriction cloning, Gibson assembly, infusion cloning, NB hi-fi assembly, Golden Gate assembly, and gateway assembly. Let's demonstrate and simulate Gibson assembly with one insert. Open the Gibson assembly cloning tool, choose the vector, and define the region for assembly. Then select the insert sequence and define the region for assembly. In the Product tab, have SnapGene design the primers required to create overlaps. Click Assemble, and that's it. We have our predicted Gibson assembly construct. We can turn on the history coloring to see the assembled fragments. In History View, we can see the steps we have simulated. The new primers are annotated on the sequence, and a list of primers is in the primers view. From here, we can export the primers and order them. So now we have our virtual construct. It's back to the lab to do this for real. Finally, you can use SnapGene to validate your clones by aligning Sanger reads to the predicted product sequence. SnapGene automatically trims low-quality regions and aligns the reads. It will automatically highlight disagreements so you can quickly jump to them. View the underlying trace and edit it if needed. You can also view the effect of differences on CDS translations. And all this work we've demonstrated is neatly contained in a single file. You can hide the alignment and return to the sequence view we had earlier, or we can jump back to the map view. The description panel allows you to add notes and other information. And that's a short demonstration of how SnapGene can improve your core molecular biology procedures and results. Visit snapgene.com to sign up for your 30-day free trial.
Okay. So now you have seen how software is used in the process of manipulating genetic molecules in the lab. So from SnapGene, you can see quite a number of things you can do, including some of the things we have seen by other software. SnapGene enables you to be able to achieve that. Now, it went even further to tell us that we can use it to do analysis of electrophoresis result. That one we are not yet used to it, but when we get there, we will be able to appreciate. So, a typical example, SnapGene enable us achieve so many things. Then the other thing that we also need to appreciate is the fact that there is what is called browsers or portals that can enable you to do further studies and research on certain genes and proteins and stuff like that. One of them is actually called Ensemble Genome Browser.
Hello everyone. In this video I will show you how to get sequence of any gene using Ensemble Genome Browser. I have already made videos on getting sequence using UCSC Genome Browser or NCBI database. So if you are interested in watching those videos, I will provide the link of those videos in the description. So let's start. So first of all, you need to open the Ensemble Genome Browser. I will provide its link in the description of this video, and you have to select the species for which you are getting sequence. So I am interested in sheep. I will click on sheep, and the gene in which I am interested is IGF-1. So I will search for it. So I will open this first link. So here is the complete detail of our gene. It is present on chromosome number 3 and it has one transcript. A gene might have several transcripts, but this gene has only one transcript, and transcript is a complete set of exons in a gene. So to get sequence, we will just click on this sequence and we will open this in new tab. But if you want to explore the transcript, we will click on this. So this transcript has 5 exons, and if you want to further explore these exons, you can click on these exons to further explore this region. So here is the complete detail of this transcript, and Here are also shown some of the variants which are reported in this gene. So if you are interested in getting details of this gene, about the variants, you can just click on the variant to which you are interested, and you can further explore it. Okay, but what for what we are interested in, we are just interested in getting the sequence of this gene. So the other tab which we have opened already, so I will go there, and here is complete sequence. So in which this first sequence is upstream sequence, and all the exons are in highlighted here. So this one is first exon, so you can check, and the other one, this one is intron and complete intron. This one is again exon, so you can find the exon or intron to which you are interested. So if you want to download this sequence, you will just click on this download sequence, and you can select cDNA sequence, coding sequence, amino acid sequence, exons, or anything you are interested. But what we are interested now is we need this complete genomic sequence. So I will click on this, and if you need any further upstream sequence, you can change it to which you are interested, or you can even change the downstream sequence if you need or not. So you will just click on this download, and this sequence will be downloaded. Okay, so this was all about today's video. Thank you for watching.
Okay. Now, what we have just seen is actually a site that you can actually go on the internet, and then you can study your nucleic acid. You can study genes. You can pick up a gene, for instance, hemoglobin gene. You want to know where that gene is actually located on the chromosome, whether it's on chromosome 1, whether it's in chromosome 2. Then you'll be able to know the number of transcript that that gene actually has. You'll be able to know the sequence. You'll be able to know the size. You'll be able to know so many things about it. And then you can compare it, for instance, if that same protein or that gene is actually found on other corresponding animals. Just like you saw in this particular example that was used for sheep. So you can check for human, you can check for chimpanzee, you can check for gorilla and stuff like that. So the assemble genome browser actually really gives you extensive information. And you also saw at the end of the demonstration, he was able to download the sequence of the gene that he was interested. So you can do so many things with that. So I will encourage you to take some time to visit the Assemble Genome website and try to see if you can download one or two or check out one or two genes and download the sequence. Thank you very much. At least that's a summary of molecular genetic tools, that we have actually just done. So what have we achieved today? We have been to achieve the fact that molecular genetic tools are tools that we can use to manipulate genetic molecules. And when we talk about genetic molecules, we are talking about the nucleic acid, we are talking about the amino acid, we are talking about the polypeptide that are going to be produced. So we need to appreciate the properties, we need to appreciate how these things are done, and what are the things we saw that we will need to use enzymes, enzyme that will cut, enzyme that will ligate, enzyme that will add, enzyme that will remove certain properties from our nucleic acid. Okay, this nucleic acid, we are going to take them somewhere to get them replicated. We will need vectors. There are different types of vectors. Some vectors are actually designed to carry very small. Some are actually designed to carry very large. We have a very large DNA inside. We should know the appropriate vector to use. Then we move to expression system. We saw the various expression system. We saw what they could do, the size, the type of, their disadvantages and advantages. We saw one by one. Then we ended up by looking at software. And then we saw different kinds of software. We now took a shot at the video of SnapGene. And we saw that SnapGene can do quite a large number of things for us. And at the end of the day, we actually saw a demonstration of Assemble Genome web browser. And like you will know, there are quite a number of other portals where you can actually look at certain things, which possibly I may add at the end of the lecture so that you can take your time to look at it. But for now, we have come to the end of today's class. So thank you very much. Except if there's any question that you will need to ask. Otherwise, we'll just close the class. Is there any question?
Okay, thank you, Prof. There's no question now, sir. The things are a little overwhelming, but you go and sit back and look at it again very well.
All right. When you look at it, you can put down your questions. So when we meet in the next class, we can ask them.
Yes, sir.
Thank you very much. Thank you very much. We come to the end of the class. So we'll close down.